So I'm Emily Gibson. I'm from the Department of Bioengineering at University of Colorado Anschutz Medical Campus. And I'd just like to thank the organizers of this great workshop. It's always really nice to hear about the latest in two-photon microscopy at this workshop. And it's unfortunate that it's not being held in Santa Barbara, especially with our record cold in Colorado right now. My talk, it's actually the same title as what I've talked about previously, but I have added some new stuff. But I'm gonna, in the first part, talk about extending two-photon microscopy to imaging and freely moving mice. And for the second part, I'll talk about some work we've done on fiber-coupled two-photon STED imaging. And so I just wanted to acknowledge, this work has involved a number of collaborators. So Diego Restrepo, Ethan Hughes, and Cell and Developmental Biology at CU Anschutz, Victor Bright, Juliet Gopinath, who are in engineering at CU Boulder, and also Mark Siemens, who's in the Department of Physics at University of Denver. Okay, so miniaturized head-attached microscopy is kind of starting to be increasingly used in sort of trying to study the brain in sort of a more naturalistic environment and freely behaving animals. And I know our last talk talked about ways of doing this sort of with head-fixed virtual reality, but in certain cases, certain types of behaviors that one would want to study, it's not possible to do this with a head-fixed apparatus. And some of the first interesting work that's come out of this was done by the Miniscope. So this was just a 1-photon wide field fluorescent image that was taken on this miniaturized microscope. And this was an example, a nice example of looking at place cells in the hippocampus and sort of visualizing the dynamics there as the mouse is navigating. Okay, so basically, when we're trying to miniaturize these microscopes, the engineering becomes a bit challenging, but we still try to do it. So on the left is kind of the classic 1-photon, wide field fluorescence microscope, where you basically have a LED light source, a dichroic mirror, and a camera. And so also what's recently being done is kind of moving this into 2-photon fiber-coupled microscopes. And so, with a nice work from Zhang in his paper showing sort of the first demonstration with this miniature MEMS device. And it's also been demonstrated with fiber scanning with a piezo as well. So what are the advantages, again, of trying to do this with 2-photon using 2-photon miniature microscopes as opposed to 1-photon? And as you probably heard a lot by now, some of the advantages of 2-photon microscopy is that you get optical sectioning images, and so you do not see the out of focus fluorescence anymore and can actually resolve cellular structures. And the other advantage is that you get much enhanced penetration depth into the tissue. So this is just showing an example of your attenuation length, at sort of 2-photon wavelengths is about roughly 130 or so microns in the mouse brain, and then recent work has pushed this even further, pioneered by Chris Chu's lab to develop 3-photon microscopy. Okay, so along with optical sectioning, it's advantageous to also be able to do three-dimensional imaging. So this is just an example of the type of images that you see and how the brain has very complicated structures. This is basically taken on an olfactory bulb. So when we were first starting to develop our miniature microscope, we wanted to find a way where we could actually image in 3D, so have a way of changing our focus into the tissue. So again, looking at some of the ways that you can do this, you've got different types of axial scanning techniques that are being more and more employed in microscopy. So one is a tunable acoustic gradient lens, which is a very fast lens that has very fast actuation rate. Also pressure-driven lens, like an OptiTune lens. And what we've decided to use is an electrowetting lens. And so the reason for that is that it is basically insensitive to vibrations and orientation, and it's very compact in size and uses low electrical power. So the electrowetting effect is kind of an interesting effect. So it was basically, first discovered about 100 years ago. If you put a droplet of water onto a hydrophobic layer, right, you'll get, you'll basically have sort of a beading happen and a contact angle. But then if that hydrophobic layer is on top of a conductor and you apply a voltage between the water and the conductor, you get a rearrangement of charge, and you can actually tune that contact angle in a very nice and reproducible manner. And so using this technique, people have generated a lot of different types of devices, such as tunable lenses, prisms, and other type of droplet rearrangements. And so you can use this electrowetting technique to actually make your own liquid tunable lens. And so this is done in the lab of Victor Bright and Juliet Gopinath up at CU Boulder. So they basically take a capillary and then coat the capillary with a conductive layer and insulating layer, and put inside the capillary like two immiscible liquids. So one is like water and oil, and then by changing the voltage right between the water contact and this electrode, you can change that contact angle. And so this is kind of what it looks like. So you get this very nice reproducible, controllable lens, and the lens can actually tune over many, many diopters, so many more than your eye can tune. Okay, and so we incorporated this into our design in our miniature microscope, so that effectively we could use this electrowetting tunable lens in our microscope to change our focus deeper into the tissue. And our design was quite simple because we basically only had a few micro lenses to basically collimate the light out of our fiber, and then our electrowetting lens, and then two lenses that make up the objective. So this was designed to effectively image through a cranial window at depths up to like 200 microns. Okay, and so this is what the design of this microscope looks like. So we've got basically the miniature head attachment that connects to a base plate. Then we have our imaging fiber bundle, and then that's attached to this adapter that actually can fit onto any type of microscope. So you can actually use this both in a confocal microscope if you want to do confocal imaging or with a modified multi-photon microscope. Okay, and so this is sort of the full experimental setup of this design. So what I'm showing over here is basically your laser scanning microscope, and that's coupling light through that objective adapter into this imaging fiber bundle, which basically relays that light onto the sample, and you use that tunable lens to adjust the depth. And so what you see out of this system is you get this kind of pixelated image because we're imaging through the fiber bundle. But then applying some filters, you can actually get comparable images, you know, to sort of a low NA objective. Okay, so, you know, using these fiber bundles works very well when we're talking about one photon imaging, but you run into trouble when you're trying to use these type of imaging fibers with intense short pulse lasers. So the first thing that happens if you're trying to use a short pulse femtosecond laser for your two-photon microscopy is that you have to deal with the chromatic dispersion of the glass in your fiber. So basically, if you send in your short pulse directly out of the laser, it passes through 1.5 meters of glass, what's going to happen is that the blue wavelengths are going to travel slower than the redder wavelengths in the spectrum, and that'll spread out your pulse in time. Okay, and so, you know, at sort of these wavelengths for two-photon absorption, you know, you have these types of dispersion curves or changes in the refractive index as a function of wavelength. Okay, and so what you get out, of course, is called a positively chirped pulse, right, where red wavelengths come first and follow the blue wavelengths. Okay, so one way to correct for this type of chromatic dispersion is to use a grading pair. So it's a way of kind of geometrically sending in your pulse. So your pulse comes in and it reflects off of these two parallel gratings and then combines and comes out. But if you look at the geometry of this, basically what happens is that the redder wavelengths will travel kind of faster than the blue wavelengths. So it kind of gives you inverse anomalous dispersion. So what you can do is put this kind of grading pair to kind of pre-compensate the dispersion of the glass in your fiber. So you send in, you know, a negatively chirped pulse into your fiber, and then you'll get out basically a fully compressed or transform limited pulse. Okay, so this one way that you can try to go about doing sort of fiber coupled microscopy is just placing, you know, this grading pair and setting it up so that you have the right spacing and it can almost balance out the dispersion that you see with your optical fiber. So that would seem to work, but actually it turns out as you try to increase your power or your peak intensity, you start to get into these nonlinear effects going on. And so what you see if you go above certain power levels is spectral narrowing. So basically, if we look here at the spectrum of our pulse, you know, it goes from its original about 12 nanometer full width half max and then narrows down to 5 nanometers. And so what happens if you have this narrower spectrum pulse just by the time frequency product, you can never get your original pulse out. So you don't have enough spectrum to get a short pulse. Okay, and so so what's happening here? So basically, when you have this kind of nonlinear effect, you have a dependence of your index of refraction on the instantaneous intensity of your pulse. Okay. And so if you look at the phase of your electric field here, so now, this is basically how we write the phase of our electric field in terms of its instantaneous frequency, and also depending on the index and the length of your material. So you can then derive, right, if you take the time derivative of the phase, that describes here the instantaneous frequency of your pulse. And what happens is this nonlinear index, right, will actually cause a frequency shift. Okay, so if we're talking about, sending in a transform limited pulse into our fiber, so this transform limited pulse has the same instantaneous frequency along the whole pulse. What happens is that due to this effect, when you have a rising edge or increasing intensity, you get this frequency shift to lower frequencies. And then at that trailing edge of the pulse, you get the opposite. And so that's why, when you send in a short pulse, transform limited pulse into a fiber with a nonlinear effect, you'll get spectral broadening. But what happens is if you send in a negatively tripped pulse, right, so if the higher frequencies are arriving before the lower frequencies, then you get the opposite effect. So you actually get spectral narrowing. Okay, so there's different ways to try to work around this so that we could increase our power further to get more efficient two-photon excitation through the fiber. And so one way to do this is to actually add in, before our grading pair, we can add in another fiber. So this is just another single mode fiber. And so when the light comes out of our laser, we focus it into this fiber, and because it's transform limited, we get spectral broadening. So then we take this broadened spectrum, send it through a grading pair, and then send it through our 1.5 meter fiber bundle. And so what's happening here again is now you're reducing that spectrum, but you can get approximately the same spectrum out to support a short pulse. Okay, so this is kind of the setup or the first setup we used to do this. So we've got our polarization maintaining fiber and our grading pair compressor. And then this is our laser scanning microscope here. Okay, so with this design, this is the kind of signal that we can see with the microscope. So this is just looking at GCamp signal through the fiber bundle. And so after processing, we can get pretty decent resolution here. Okay, and let's see. Okay, and because we have that electrowetting lens in there, we can actually scan through the sample of about 200 microns. And so that allows us to visualize sort of different structures. Okay, and so these are the measurements that my graduate student Barish Azbeh did. So this is just looking at signals that were taken in 3 different layers. Okay, and so this is just kind of showing, if you have the mouse in a cage. So this was doing some imaging of our microscope, actually going through an implanted grin lens in the mouse to the piriform cortex. And so the mouse is able to kind of freely move about the cage. And we see this correlation between when it gets close to some novel bedding, we see activity in piriform cortex. Okay, and then some of the other advantages, since you are using sort of a standard microscope, you can actually do multicolor imaging. So this is using our miniature microscope, we can get images of cells that are labeled, right? One type of cell with GFP and the other with TD tomato. And the other thing that's kind of nice, so when you have this voltage tunable lens, you can actually change the plane that you're doing your imaging. So you basically, as you're scanning along, as you go on the y-axis, you can change the voltage of the lens and then scan at a tilted plane. And so again, that might be an advantage if you don't happen to insert your or completely align your microscope onto the sort of region that you want to look at. Okay. So what are some of the issues with trying to do two-photon microscopy through fiber bundles? So, these types of fiber bundles are or coherent fiber bundles are nominally used sort of in one-photon endoscopy applications. And so you get nice uniform transmission of fluorescence if you have one-photon excitation. But if you have two-photon excitation, this is kind of what you'll see. So you get these kind of really sort of inhomogeneous signals. And this is just looking at a uniform fluorescence slide. This is imaging through a uniform fluorescence slide with two-photon. So we've kind of gotten around that by taking sort of calibration measurements of the uniform slide and then trying to correct for that in our imagery construction. So other issues, that you run into with these fibers are you're kind of limited in sort of how closely spaced your cores are, because if you try to go closer, you can get core-to-core coupling between different fiber cores. So with the one that we use, we don't see a lot of like core-to-core coupling, but you can see these kind of additional higher order modes getting translated to other fiber cores. Okay, and so this is kind of, so showing on the right, this is an example like of a cross-section of this kind of Fujikura imaging fiber bundle. So it's an SEM image, so you see that, right, these cores are not really uniform, they've got varied shapes and sizes. And so what you end up seeing, when you're trying to do two-photon imaging is that, right, due to the variations in geometry or possibly dopant material, different cores will be bright depending on different grading spacings. So now you've got, these cores and they've got varying levels of dispersion, and you're trying to compensate for that. So these are definitely all things that kind of need to be worked out, I think, when we're taking this further. But again, just to emphasize, some of the advantages of this type of microscope, when we're using a fiber bundle, basically kind of the nice thing is that, the head attached part can be much simpler, much easier to assemble, and lighter weight, because you don't have this like lateral scanning device. And then one of the other advantages is that you could potentially use the same device to do optogenetic stimulation. So you can actually illuminate different fiber regions and do stimulation. So again, I think that these are really interesting advantages in finding ways to improve uniformity of our transmission is important. Okay, let's see. So the next part of my talk, I was going to talk about some recent work that we've done in collaboration with Juliet Gopinath and Mark Siemens. So this was work on developing a miniature fiber coupled STED microscope. Okay, so we thought that, doing 2-photon miniature microscopes wasn't hard enough. So we said, let's try to develop a miniature STED microscope. And so surprisingly, NSF gave us some money to do it. Okay, so, some of the interesting things, if you can actually achieve this kind of high risk goal, is that you could actually, image, important subdiffraction limited structures in the brain, in sort of naturalistic behavior, or also do kind of long-term longitudinal imaging. And so some of the interesting structures you can look at are, dendritic spine morphology or protein localization. Okay, so STED. So basically, STED or stimulated emission depletion microscopy is the technique that was invented by Stefan Hell, or first demonstrated by Stefan Hell. And so, traditionally, microscopes have been limited, of course, by the Abbey limit, right, which is how tightly we can focus our light through a certain numerical aperture. And so with kind of the resolution you get from a standard confocal microscope, right, we're limited by the wavelength over 2NA. And so this is as tight as we can focus. So we could really only see, we can't resolve individual fluorophores or structures because we get this blurry image out. So with STED microscopy, so what Stefan Hell did was take a beam that was shaped into this donut at a different wavelength. So it's at the wavelength of the emission of the fluorophore. And what happens is that in the region where you have this intensity in the STED wavelength, you actually get stimulated emission of those excited fluorophores. And so those effectively turn off and you get a much smaller point spread function out. Okay, and so this is the equation for your resolution, which depends on the intensity of your STED light divided by the saturation intensity. Okay, and so you can also do 2-photon STED. So if you want to actually image, in vivo in tissue, people have demonstrated this on benchtop systems. So you use your 2-photon excitation source, and then have sort of the same wavelength depletion source. And so with this technique, it's been sort of super resolution has been demonstrated. I think this might be the record. I'm not 100% sure, but 76 microns deep in mouse brain. Okay. And so again, so you have the same process, basically you have 2-photon excitation. And when you apply your STED beam, then you get depletion. Okay, so we wanted to, in order to try to develop this in head attached microscope, we need a way of sending sort of this donut beam through an optical fiber. But the problem is, these beams are not stable in most types of optical fibers, although it was demonstrated by a custom fiber that did support a donut mode. But so our approach was to use basically a commercially available polarization maintaining fiber. And so what you do here is we kind of launch these two different fiber modes, mode 1 and mode 2. And so if these are like offset by you know, a phase of pi over 2, then you'll generate this vortex beam or donut mode. Okay. So that's fine. You can do that in a fiber, but the problem is if you bend the fiber at all, you're going to change the phase between those two modes. So then it becomes kind of pointless if you're trying to use it for head attached microscopy. So how could we solve that problem? So instead, the idea that Mark Siemens came up with was, why don't you use two incoherent modes? So we basically delay those two modes in time. So you're beyond the temporal coherence. And then if you bend the fiber, it doesn't matter. And then the summation of those two modes will generate this donut mode. Okay. Okay, so this is the experimental setup. So this was developed by Brendan Heffernan, who is a graduate student that recently graduated from Juliet Gopinath's lab. So he generated or launched these fiber modes and kind of used a dove prism and a delay line, so that they were separated out and incoherent from one another. Okay. And so, or I should say, sorry, going back. So, so these two modes were launched into the fiber along with, short pulse at 900 nanometers. Okay, and so after that, you can basically take the output of the fiber and then focus it down and perform STED with this, which is great. So this is looking at sort of the images that Brendan got with like 100 nanometer bead sample. So it showed almost a threefold improvement in the resolution. And then this is showing, some images that he took of mammalian cells. So this was immunostained with a barrier star green dye, which is a good dye for STED. So saw definite enhancement in resolution there. Okay, so some of the things then like we noticed happening here. So so it turns out, okay, that right, the polarization of this kind of donut mode has a pretty strong effect on so your depletion, your STED depletion efficiency. Okay, and so what happened here, it seemed to be a combination of both sort of the polarization dependence of transition for two-photon excitation, as well as the efficiency of your STED depletion. So on the top here, so this was basically sending in light where you had like one polarized excitation, linear polarized excitation. And then, so these donut beams had to be polarized this way. So they had to be, one mode had to be polarized like this and the black arrows, and the other had to be the white arrows. Okay. So what happened here, right, is that basically you form a dipole, or more of your dipoles are aligned along the red direction, and then you only see more enhancement, right, along the region that's at the same polarization.
Emily, can you
Yeah.
unpack why the two depletion beams have to have opposite polarization? I guess they're being combined by polarization, but it's probably something else, right?
Yeah, exactly. So, so that is what happens here. So it's basically due to the fact that, so if you've got like radial polarization, which is kind of what you would want, if you put that in type focusing, then it'll actually, you won't get that null in the center. So you're kind of forced to have polarization in this direction, so that when you're using a type focus like a 1.4 NA, you'll still get that null in the center. And so that's why you have to use circularly polarized light sort of for STED in the traditional way. When you're using the vortex phase, it has to be circularly polarized. And if it's even slightly not circularly polarized, then you'll get some intensity in the null of the donut, or the donut hole, whatever you want to call it.
Super clear, thanks. I should have just waited a slide.
So anyway, so this was kind of an interesting effect. And then also, so Brendan tried putting in sort of two excitation pulses at orthogonal polarizations. And so what you see here is kind of a more uniform, a uniform efficiency, but you still have, just due to the polarization, you still have some percentage of the dipoles that aren't sufficiently depleted. And so you do get this kind of background pedestal. Okay. But yeah, so anyway, it was kind of interesting to like see this play out. And the fact that, going back, it turns out, right, the fact that the single photon transition doesn't depend on your polarization of your excitation light, but two photon does depend on the polarization, and the degree that it depends on it depends on the molecular structure of what you're looking at. So for example, GFP, I think you have 30% less efficient transition if you're using circularly polarized light versus linearly polarized light. So that's something that, I didn't kind of know at first when I was doing two-photon microscopy. Okay, so that was all I wanted to talk about. I just want to acknowledge all the people that were involved in the work. So mainly Greg Futia and Barish Osbey in my lab did the work on the miniature two-photon electrowetting microscope with the help of Omkar Supakar, who's in Juliet and Victor's lab. Brendan Heffernan is a graduate student that recently graduated from Juliet's lab and was helped by Stephanie Meyer, who's built some custom STED microscopes in our lab. And so that was basically it. And again, we're always looking for interested, motivated grad students and postdocs who want to join our neurophotonics group in beautiful Colorado, where there's beautiful hiking and skiing and rock climbing. Thank you.